right, so for our science fair, we're testing the effects of UVC light on bacterial growth.
So what's the big deal about UVC light? Well, there's a couple reasons. First of all, it's more sustainable than classic cleaning products. Like, there's no waste, so you don't have to be throwing away wipes and bottles that cleaning products normally come in, and instead you just flip a switch, the light turns on, and kills all the bacteria. Second, it's safe on surfaces, so it doesn't leave behind the chemical residue that most cleaning products leave behind, and instead, again, it's just a light and it just kills the bacteria and leaves the surfaces undamaged. Also, there's no fumes. So, unlike cleaning products that smell like chemicals or smell sometimes like alcohol, the light does not have a scent. Also, it's more cost efficient. For example, there's less transportation involved, and you don't have to have trucks and shipments of bottles of cleaning products, and instead, it's just a light that you switch on. And lastly, there's applications in robotics that would make it more efficient than janitors or people to go around cleaning things, and you just have robots move around with the UV lights.
So how does UVC work? UVC light affects bacteria by damaging and altering the bacterial DNA. So it's considered actinic, which means it has enough energy, which is like demonstrated by its wavelength, to go inside the cells and alter the cell's DNA. And when it does this, it forms structures called dimers, which is between two adjacent thymine bases. It'll form a double bond, and this double bond will prevent the cell from doing important things like binary fission, so the cell can no longer reproduce. Also, when these dimers are created, the bacteria will try to repair it, and sometimes mistakes will occur, which will cause further problems in the bacteria. Um, and yeah, again, these mistakes can even kill the bacteria cell or prevent binary fission, which will basically deactivate the bacteria. And here's a diagram. As you can see, two adjacent thymers right here that are double bonded.
Okay, so our experiment focused on how time affects UVC disinfectant. So the number of dimers produced in the bacteria increases with increased time of exposure to UVC light. We are trying to figure out how quickly can UVC light create enough dimers to kill 99.9% of bacteria, which is what most chemical disinfectants advertise that they are able to do. And with that, we are trying to decide whether or not UVC light is a viable solution to chemicals and what practical applications it might have in a professional setting.
So our hypothesis is if longer UVC light exposure time creates more thymine dimers in bacteria, then bacteria with a longer exposure period under UVC light will have lower colony counts after growth. So here's our experiment. First, we prepared petri dishes. So we got agar that would be suitable to grow the bacteria, and we plated it in the dishes. And then we had to collect the bacteria where we got from communal school computers. We were trying to find common bacteria, so that's where we went. Then we put these dishes under UVC light for different time periods, 15 seconds, 30 seconds, a minute, and then one group set aside as a control with no light exposure. We incubated these dishes for 48 hours, and after 48 hours, we counted the number of colonies present. One thing to note is UVC light is dangerous if it's on your skin for extended periods of time. So we put our light inside a cardboard box to shield the light from hitting us as we were experimenting.
Okay, so here's some pictures of what our petri dishes looked like after they were finished growing and inside the incubator. And so you'll see in the top left, we have our group with no light exposure. And you'll see that there's a lot of colonies, a lot of large colonies too. And if you look at the bottom right at 60 seconds, and even if you look at 30 seconds, there's a lot less colonies. You'll also notice that each group, like each petri dish, is labeled with its amount of time exposure along with the letters so we could keep track of which trials were where. And also, you'll note that there's little black dots on each petri dish, which is how we were able to count the colonies. and collect our data.
All right, so here are our results. Um, After averaging all of the colonies together for each trial, we put them on a graph and found the line of best fit. And we found that time and bacterial death is not actually proportional. It's actually a natural log function. And we found this by finding the highest R squared value for different types of functions. And we found the highest one is with the natural log function. Yes.
This data illustrates that more time under the UVC light decreased the bacteria growth because of the UVC radiation, which supports our original hypothesis that increased time exposure to UVC light will decrease the number of colonies per petri dish. And we also found out that the rate that bacteria is killed decreases over time, which was an interesting thing that I think we both learned from this experiment. Then we also had to decide, again, like how long is UVC needed to kill 99.9% of bacteria, as that's what most classic cleaning products advertise they're able to do. And if we use the equation that we found based on our data, we found that to have 0 colonies, you need to have UVC exposure for 333.7 seconds, or about 5 minutes and 34 seconds of exposure.
All right, so there was a couple questions posted, and we wanted to answer those. So the first one was, what interests you about this topic, and where did you get the idea for your project? So I know recently COVID has caused a lot of demand for cleaning products, and of course bacteria are different than viruses, but we weren't able to test viruses, so we decided we would test bacteria instead with UV light, because UV light could be a better alternative to cleaning products with the things we discussed earlier, talking about having enough of it and not running out, and like not needing someone to apply the UV light. It can be done by robots, and but we wanted to also know, like, is UV light viable? Like, could you use UV light where it'd kill the bacteria slash viruses in time? And with our experiment, we found that in about 5 minutes, it effectively kills bacteria.
Another question was, what did we learn about the science behind our project before and during the experiment? So I know one thing that I think we both learned how to do is how to plate agar into like petri dishes and take bacteria samples and just some of the like gritty stuff of the procedure.
Yeah, and certainly about how bacteria works with the DNA, and how UVC light works with the double bonding on creating dimers between the thymines.
And even just the like simple like cellular structure of bacteria and how it's a much more simple it's a much more simple being than humans and like human cells.
Definitely, yes. And then if we had to do it again, what would we change, and what improvements would we make?
Yeah, so a couple of things went wrong in our experiment. We actually had to do our experiment twice because the first time all of our agar dried up, which meant that in order to save time on materials and save some money, we just used the same agar plates, which was meant that the agar plates weren't completely sterile. Although we did bleach them, they weren't, brand new out of the box, which could have affected our findings. So if we did it again, we probably would need to use new plates. Another thing that went wrong was some of our dishes had developed like a lot of air bubbles. And I'm not really sure how that could be fixed, but maybe if you have enough trials that you're able to like discard them from your data set to make sure that you have accurate data. Another thing that happened was when we were swabbing, we swabbed from computers, and there was really no way to tell if we were having equal amounts of bacteria on each plate to be developed. So I mean, it was as like accurate as we could. It was like a couple swipes on each dish, but again, like we had no way of knowing that there was exactly equal bacteria on each plate. So maybe a way to fix that could be buying bacteria from retailers and measuring the amount of bacteria you're putting on each plate.
So what's next? Continue the project. Go on to a different topic. So the thing we were talking about was like applications of this UV light. So there's a couple that are actually already in use, but UV light in air filtration systems, milk pasteurization, water purification. We could test UVC light's effectiveness in all those areas and find, like what can we use this UVC light for. Um, Other things, like the things that affect the rate of UVC lights, like effectiveness. So like how strong the light is, the concentration of the light. Also shadows, like how will shadows factor in to affect the UVC light's effectiveness. All of those things are things we could do for what's next when we continue this project. And bibliography.
Okay. Thank you so much for listening to our presentation about the effects of UVC light and how time plays into it all. And I'm Sarah Driscoll,
And I'm Garrett Manister.
and have a great day.